Next, I want to talk about some of the patterns of inheritance that we might see that don't follow this complete dominance pattern, so where the dominant always masks the recessive. One example of this is what we call incomplete dominance, which is where we see that the phenotype of the offspring is going to be somewhere in between the two parents. And the typical example that's given here is that the homozygous red parent, which is dominant, the homozygous red flower, mates with a homozygous recessive white flower. And the offspring are going to be a pink condition. So that heterozygote individual is actually going to be pink. And so the offspring is going to have an intermediate color between the two. Another thing that you might see, another pattern of inheritance, is what we call codominance. And this is illustrated by the blood groups in humans. And so blood groups in humans, you can be of several different phenotypes. And what we have with this codominance is that, in fact, the heterozygote condition is going to show both of the traits of that allele, both the recessive and the dominant trait. Okay, so what do I mean by this? Well, there's a variety of phenotypes of the blood groups for humans. You can either be phenotype A, B, AB, or O. And all of those are going to correspond with what the markers on the surface of the red blood cells look like. So if you're phenotype A, you might have, just like you can see in this diagram here, little triangles on the cell on the outside of your red blood cells. If you're phenotype B, you might have little circles. If you're phenotype AB, you, because of this codominance, are going to have both circles and triangles. And if you are O, you're not going to have any markers on the surface of your red blood cells. Now, when we look at the genotype, there's a couple of different ways that you can achieve some of these phenotypes. For example, if you are blood group phenotype blood group A, you can either be a homozygous for the A condition, so you can have two A alleles, or you can have an A allele and an O allele. For the B phenotype, you can have either two Bs, be a homozygous, or you can have a B and an O. That will still make you a B phenotype. If you have an A allele and a B allele, you will be blood group AB. That is the only way that you can achieve that phenotype. And the only way that you can achieve that phenotype for blood group O is to have two O alleles. Okay? So let's take a look at some examples that you might encounter in your problem set. All right. In cattle, polled horns, which is short horns, is dominant over long horns that are normal. A separate gene for coat color has two alleles that can result in three different phenotypes. RR is red, WW is white, and RW is roan, where the red and white are equally expressed. What kind of dominance is demonstrated for the coat color? Well, here the key words are that red and white are equally expressed. And if they're equally expressed, that means that you are looking at codominance, just like those blood groups. All right, let's keep reading here our question. So we have a bull who is who is a roan polled homozygous bull with a roan long-horned cow. That's what our mating event is going to be. If the traits are unlinked, which the question is telling us here, we have no reason to believe that they might be linked. We're going to talk about that in just a little bit. What proportion of the offspring would you expect to be red and polled, roan and polled, or white and horned? So what we're going to do here is take a look at what the two different Punnett squares for both horn and coat color tell us. Let's start with looking at the coat color. So we have a roan bull. If he is a roan bull, we know that his genotype is RW. Now, this bull is going to mate with a roan cow. So we know that her genotype is going to be RW. So we can fill out our Punnett square here. Big R, big R, big RW, RW, and WW. And we can get our phenotypes. 1/4 is going to be red. 1/2 is going to be roan. And 1/4 is going to be white. Okay. Now let's take a look at the next trait that we're interested in, and that's the horns. So we know that we have a polled bull, and that the polled bull is going to be homozygous, and that is going to be the polled horns, which are short, is going to be dominant over the normal horns. So we can say that the male who is polled is going to have 2 dominant for that, and is going to mate with a long-horned cow. Okay, the only way that a cow can be long-horned is if she has 2 recessive alleles. So we can fill out our Punnett squares. All right, so now we can figure out what our expectations are here for the offspring. So, what proportion of the offspring would you expect to be red and polled? So the probability of being red is 1/4, and the probability of being polled is, well, there's only one type of offspring that we're going to get for that. All of the offspring of this event is going to be polled, right? Because they're all going to have 1 dominant allele. So that's times 4. It's going to be 1/4. What is the probability of being roan and polled? Well, that's going to be 1/2 times 1. And finally, what's the probability of being white and horned? So the probability of being white is 1/4. What is the probability that you are horned? There is none. You only have a probability of 1 for being polled, so that is 0. That means that the probability of getting white horned offspring is 0.